Viroids are the smallest known plant pathogens, consisting of small, non-coding, single-stranded enclosed RNA molecules. Despite their simplicity, they are still able to infect a host cell and spread systematically in the plant and thus cause a lot of diseases. Viroids are important because they cause a lot of yield losses in important crops such as tomato, potato, citrus, and so on. We still don't have effective control strategies to deal with viroids. So my name is Teodora and I'm a part of ViroiDoc. We are a doctoral network of PhD students and all of us focus on addressing the challenges posed by viroids. My project in particular focuses on developing new antiviroidal strategies, meaning that we are trying to get one step closer to an antiviroidal drug discovery. I am working under the supervision of Professor Purificación Lisón at the Polytechnic University of Valencia. And during my project, I will also have secondments at the University of Crete in Greece under the supervision of Kriton Kalantitis, and also in Bari in Italy under the supervision of Francesco Di Serio. To do this, we will engineer B-White2 tobacco cells, which will constitutively express citrus exocortis viroid or potato spindle tuber viroid. This will allow us to create a robust model for screening of potential compounds that can be used against viroids. So one of the compounds that we are starting off with, for example, is salicylic acid. Salicylic acid is an endogenous plant hormone that is fundamental in the signal transduction pathway of plants, meaning that it is really involved in plant stress response against biotic and abiotic stress. For example, transgenic tomato plants, which have been engineered to not be able to accumulate this hormone in them, show extreme hypersusceptibility to the citrus exocortis viroid. This shows that salicylic acid is directly correlated with plants' response to viroids, and this is why it's definitely on our list of candidate molecules to screen. Gamma aminobutyric acid, or also known as GABA, is a functional metabolite that increases in tomato plants once they were treated with benzothiazole, which is an analog of salicylic acid. GABA helps plants resist citrus exocortis viroid by delaying plant symptom development and also even reversing susceptibility in plants that cannot produce salicylic acid themselves. It also activates several plant defense genes, making it a very good potential antiviroidal candidate. Ethylene is another plant hormone that plays a role in viroid disease. In tomato plants infected with citrus exocortis viroid, it delays plant symptom development and at the same time reduces ribosome stress. Another candidate molecule is AZD8055. This is a compound that's an inhibitor of the target of rapamycin pathway, also known as TOR pathway. Autophagy helps plants fight off viroid infection, reduce viroid levels, and even make symptoms like stunting go away. This target of rapamycin inhibitor also boosts the plant's immune system, making it a very good candidate for antiviroidal screening. We will be using methods such as reverse transcription and quantitative PCR. We are using fluorescent microscopy to do subcellular localization. We are using small RNA sequencing. In addition, we will be also analyzing the expression of host marker genes such as PR1 and NAC82. The promising compounds that we will screen will be then subsequently tested also in planta. We will be testing on tobacco plants infected with potato spindle tuber viroid, and we will also be testing on tomato plants infected with both potato spindle tuber viroid and citrus exocortis viroid. After this, we will also extend our study to another family of viroids, the afson viroids, and we will be testing on plants infected with chrysanthemum chlorotic mottle viroid. This research and my project in general is bringing us one step closer towards new antiviroidal drug discovery, which can be used as an effective control against viroids and help in agriculture.